Department of Microbiology. Bill and I share common roots. We both received degrees from that great university of the heartland. Bill receiving his bachelor's degree and I my PhD degree. Bill is, apart from the science, I'll get to that in a second, extremely well known for his ability as an instructor. In fact, I see is consistently one of the excellent instructors on the U of I campus. He's been co-director of the Woods Hole Microbial Diversity course, which is one of the premier courses internationally recognized. Co-director of the Cold Spring Harbor Advanced Bacterial Genetics course. You should expect a good talk today, I think. So the science. Bill has taken two trajectories, one towards carbon and one towards phosphorus. And today you'll hear about strange redox states of phosphorus you probably haven't thought much about, and novel compounds that contain those phosphonic acid residues. The other part of his lab is focused on methanogenesis. So these are really totally disparate research. Well, they are disparate, but I'll give you a heads up that today, Bill, I think, is going to bring these two disparate lines of research together in an interesting environmental story. Without saying anything more.
So Dave, thanks for the invitation and for the nice introduction. What happens if I give a bad talk?
I'll hand it out. You got it.
Okay. Okay, that's good. So everybody can hear me? All right. So what I'm going to do today is tell you some work that we've been doing at University of Illinois towards the discovery and the development of novel antibiotics and also of known antibiotics. And I think before I start, I want to make sure that I give credit where credit's due. Most of what I'm going to talk about today was done in my lab, but I couldn't do the work I do without the help of some very talented collaborators, including Wilfred Vanderdonk in the Department of Chemistry, who's a great synthetic chemist and also a mechanistic enzymologist. Huimin Zhao in the Department of Chemical and Biomolecular Engineering, who's very good at synthetic biology and strain engineering approaches. Satish Nair, who's a protein crystallographer, and Neil Kelleher, who sadly has recently moved to Northwestern. Sadly for us, I mean, it's a great plus for Northwestern, who's a very talented mass spectroscopist. So let's start just by introducing the topic. And I think in a microbiology seminar, everybody's familiar with pathogenic microorganisms, and I think everyone is familiar with the danger that's posed by antibiotic-resistant pathogens. So we now are at a day and age where many fairly serious pathogens, things like tuberculosis, are resistant to many of our antibiotics, fast becoming resistant to some of our very best and our newest antibiotics. Actually, typically an antibiotic is on the market for something like 6 to 12 months before you have the development of clinical resistance to those. So antibiotic-resistant pathogens are a huge issue. We've all been really lucky. We've lived our lives in kind of the golden age of antibiotics. That's sort of illustrated on this slide, which shows the discovery of the first antibiotics back in the 1930s. And there was this great wave of discovery of new antibiotics, which sadly the pathogens become resistant to almost as rapidly as we develop them. So that's fine as long as you keep developing new antibiotics. Hopefully somewhere along the line we're going to become a little bit smarter about the way we use antibiotics and hopefully curb the spread of new resistances. But in the meantime, the discovery of antibiotics has tapered off rather dramatically, such that in the last decade, only 5 new groups of antibiotics have found their way onto the market. So this is kind of an alarming trend. More and more resistant pathogens, fewer and fewer drugs to treat them. So what are we going to do about this? So you can ask yourself a question. Why are we discovering fewer and fewer antibiotics? I can think of 3 reasons. 1, we stopped looking. 2, there are no more. 3, we're looking in the wrong way. So I can tell you that 2 of those 3 are true. Pretty much we have stopped looking. So the large pharmaceutical companies, the majority of them, have completely eliminated their antibiotic discovery programs. It simply costs them too much money relative to the amount of money that they feel they can earn back from it. They would much rather see you take a cholesterol medicine that you would have to take every day for the rest of your life than an effective antibiotic that you'd take for 7 days and be done with it. So a lot of the pharmaceutical companies have stopped looking. So that leads, there are no more, or we're not looking in the right way. So there's lots of evidence, and I'll show you some of that evidence, that there are plenty more natural products to be discovered. What's going on, I think, is that we're looking in the wrong way. So this has been a very effective strategy for nearly half a century, but what we're finding today is the same things over and over again. And I think the reason for that is that we're looking in the wrong way. So traditionally, the way this has been done is that people would go around the world and they would collect microorganisms. The vast majority of antibiotics actually come from a single genus. Something I learned recently, I don't know how many of you know this, 70% of pharmaceutical treatments, I'm not talking about antibiotics, I'm talking about all pharmaceuticals, come from a single genus, Streptomyces. They're either directly produced or they're chemically modified derivatives of things that are produced by these microorganisms. So the best place to look for antibiotics is in microorganisms. They use them, depends on who you talk to. They use them either as chemical warfare to kill off the competition. Others would tell you they use them as signaling molecules to talk to the other microbes that are in the environment. So if you think about the way this has been done over the years, you go around the world, you collect microorganisms, you get it in pure culture, you bring it in the lab, and you grow it up, and you expect it to produce some compound that will kill some other organism. So they have to produce it in high enough concentrations that it'll kill this other organism in some kind of panel of test organisms that you're screening. So think about this. If it's for chemical warfare, and you brought it in pure culture and you're growing it in the lab, who are you going to kill? Or if it's for communication, who are you going to talk to? The truth is, the organisms that make these spend a fair bit of their cellular resources in synthesizing these antibiotics. Why would they do that? Produce a compound that they clearly don't need in pure culture in the lab. And the answer is they don't. So the vast majority, the vast genetic potential for natural product, you can read that as antibiotics, produced by microorganisms is not accessed by growing them up in pure culture in the lab. Let me give you some evidence that that's true. So here's one of my favorite organisms, Streptomyces silicolor, sky-colored. I wish I had taken this picture. This is a David Hopwood picture. It's one of the best microbiology pictures I know of. So this is an organism that's been intensively studied for about 60 years. And in 60 years, we discovered that this microorganism produces these 3 antibiotics, and it also produces this natural product, which is the compound that gives soil that earthy smell. So if anybody comes in my lab and they say, it smells just like fresh dirt in here, I say, no, you've got that just backwards. Fresh dirt smells like Streptomyces, because that's how many of them are living in the soil. So 60 years, we discovered these 4 molecules. About it, I guess almost 10 years ago now, the genome of this organism was sequenced. And it turns out if you look at the genetic complement, it has gene clusters for the synthesis of 24 natural products. So in 60 years, we found 4, and it has an additional 20 that it produces. These are not just genes that happen to be on the chromosome. Knowing that these gene clusters are there, if you go back and look with highly sensitive detection methods, these code for the products of natural products, some of which are antibiotics. So we're not accessing about 90% of the genetic potential of this microorganism based on its genome sequence. So we now have done the genome sequences of about 20 Streptomyces. This story is repeated in every one of them. So your average actinomycete has natural product biosynthetic capacity to produce 20 to as many as 48 different compounds, many of which are antibiotics, many of which are useful pharmaceutical compounds. So there's a great wealth of information, genetic capacity to make new natural products if we could only tap into it. My feeling is that nature's been doing this experiment for 4 billion years. If there's a valid biological target, nature's discovered it, and we simply need to tap into it and figure out what's there, bring it into the lab, and figure out what they're doing. So the approach that we've taken is rather than grow the organism and look for specific activity, we look for the genetic potential to make one of these natural products. And then we bring it into the lab. And I'm a molecular biologist, a geneticist, microbial geneticist. I don't care if the organism wants to make it, I insist. We can change the promoters, we can rig the growth conditions, we can get them to express these genes. We can purify the compounds, we can get them in large amounts, and then we can run a panel of bioactivity testing and use that approach to find the compounds that nature has already discovered. So if you're going to do that kind of gene-based approach, you'd like to pick a class of molecule that has a potential to be bioactive. So the class of compound that we've chosen to look at, as they've suggested, are phosphonic acids. So this is where carbon meets phosphorus. So the diagnostic trait of a phosphonic acid is it has a direct carbon to phosphorus bond. And what I'm showing you on this slide are about a dozen phosphonic acids that are natural products. So it turns out lots of microorganisms, lots of so-called lower eukaryotes, things like mussels, sea anemones, corals, produce this molecule, aminoethyl phosphonic acid. And what they do with that is they make phospholipid. So instead of making phosphate ester-linked lipids, they make phosphonic acid-linked lipids. They also make exopolysaccharide that's associated with this molecule to give it a little bit of negative charge, which they put out on the surface. And they use it for this kind of structural purpose. The idea is that the carbon-phosphorus bond is very stable, and if you're going to put a valuable resource like phosphorus on the outside of your cell, you'd like to put it in a form that it can't easily be stolen away by other organisms. So lots of organisms make this, and an organism like sea anemone, half the phosphorus in the organism is in the form of phosphonate, as opposed to phosphate esters, which I think most of you are more familiar with. The remaining compounds are bioactive molecules. Most of them are antibiotics. And they're a class of antibiotics that target a wide range of activities. So in order to understand that, I'm going to show you why these work as antibiotics. So phosphonic acids act as chemical mimics. So you'll notice across the bottom here, I have 4 different metabolites that are important for lots of living organisms. And across the top row, I have a series of chemical analogs, which instead of having this phosphate ester linkage, have the phosphonic acid linkage. So that's true for these two. Or instead of a carboxylate group, they have a phosphinate or a phosphonate moiety, which due to its charge density actually is a pretty good mimic of the carboxylic acid. So the thing about carbon-phosphorus bond, as opposed to the phosphate ester linkage, is that it's incredibly stable. So these are labile linkages. They're easily acted upon by enzymes. The carbon-phosphorus bond is extremely stable. So most of these molecules you can boil in 10 molar acid or base for a week, and they'll be stable. Nothing happens to them. So what happens, though, is you notice that the chemical structure is very similar. So if you have an enzyme that would bind this intermediate, very likely it also binds this intermediate. Chemistry can happen here. Nothing happens here. And if it binds the enzyme very tightly, it inhibits that enzyme. If that enzyme is important to a biochemical pathway, that biochemical pathway is blocked. And if that biochemical pathway is essential to the organism, you've killed the organism. So what you're looking for is a target that will affect a biochemical pathway that's found in the microorganism that's not found in the human beings. So this is an intermediate in threonine biosynthesis. Human beings don't make threonine. You have to get it in your diet. So this is actually a target that's threonine synthase that's absent in humans, that's present in bacteria, it's present in fungi. And this is a very good antifungal and antibacterial agent that's utterly non-toxic to humans. This one inhibits the non-mevalonate pathway of isoprene biosynthesis. This one inhibits glutamine synthetase. This one inhibits alanine racemase. In all cases, these are targets that are important to the microorganism, but that are not found in humans. So as I said, you want to pick a group of molecules that has a range to target, that has the capacity to target a wide range of different cellular targets. So I asked people to think back to all the biochemical pathways you had to memorize in biochemistry or in your microbiology class. And I want you to ask yourself, how many pathways did not have at least one phosphate ester or one carboxylate? I think the answer is 0. So what that means is that there is potentially a phosphonate inhibitor for every biochemical pathway in nature. That's a very powerful class of molecules. I would go even further than that. There are many of these molecules They don't necessarily even have to kill. So for example, this molecule, K26, is a peptide mimic that inhibits angiotensin converting enzyme. That's a protease that takes pre-angiotensin and turns it into angiotensin, and that's how your body regulates vasoconstriction. In other words, it regulates your blood pressure. So this is not an antibiotic. This is a molecule that is every bit as good as anything used to lower blood pressure. There are others that inhibit phosphorylation of molecules that regulate cell division. In other words, they're anti-cancer agents, anti-neoplastic agents. So you can inhibit regulatory phosphorylation or regulatory dephosphorylation. The range of targets hit by these phosphonic acids is huge. So there are known antibacterials, known antivirals, known anti-HIV protease inhibitors. Herbicide Roundup is a phosphonic acid. So it's a really good class of molecules. As I said, all of these were discovered in natural product screens that were looking for the activity. We would like to use our gene-based screening approach to see, number one, how are these molecules made? And number two, are there other phosphonates that nature has discovered that haven't been looked for, or that haven't, I should say, been discovered by using this traditional screening approach? And I'll show you, I think, why we wanted to do both of those. So the way you do a gene-based screening is you have a biosynthetic step that's important for the synthesis of these molecules. So it turns out, with a single exception, which happens to be this angiotensin converting enzyme, all known phosphonates share this first step in their biosynthesis. So they have a common step that's required for all of their biosynthesis. And that's the installation of the carbon-phosphorus bond by rearrangement of phosphoenol pyruvate to phosphonyl pyruvate. That's a very endergonic reaction. In fact, the equilibrium is far in this direction. So the cell has to couple that with an irreversible reaction to pull this towards net synthesis of phosphonic acid. And the enzyme that does that is called phosphonyl pyruvate decarboxylase. So when we started this, we targeted both of these genes as potential gene-based screens. It turns out that this one works really well. This one doesn't work so well because it turns out nature has discovered other driving reactions to pull this away and make the net synthesis. So enough PEP mutase genes had been discovered over the years that we could do an alignment and we could develop degenerate PCR primers so that we could amplify the PEP mutase for most organisms that had the genetic capacity to make a phosphate. So if you have this gene, you have the genetic capacity to make a phosphate. Over the years, we've actually refined those primers, and we now have a set of 4 different primers that will amplify the PEP mutase gene from every single organism that's known to have it. So we think we've developed a pretty good gene-based screen for the genetic potential to make phosphonic acids. All right. So the next thing I need to tell you is how friendly antibiotic biosynthesis is for the molecular biologist. So most microorganisms that make natural products, even when the biosynthesis is fairly complicated, it might require a pathway of 10, 15, even 20 different steps, each of which is encoded by a different gene. Typically, they put all those genes together in the same place on the chromosome. So if you can clone any one gene in the pathway, you can clone all the genes in the pathway. So what we do is we make large insert cosmid clones from an organism that we've shown by our gene-based screening has a PEP mutase. We then screen the genomic library to find the clones that have the PEP mutase. And we sequence those, and we can typically get the entire antibiotic biosynthetic gene cluster in one cloning step. Here's an illustration of that. So these are 3 different organisms that produce a phosphonate antibiotic called phosphinothrycin tripeptide. If you're from a farming area, you might know this is the herbicide Liberty. It's a widely sold herbicide. We cloned the PEP mutase gene from all of these, and we were able to clone 40 to nearly 50 kb gene clusters that encode the biosynthesis of this antibiotic. How do I know that we cloned the entire gene cluster? So we can take those clones. So here's an organism that produces PTT. Not only is it a good herbicide, but it's a good antibacterial compound as well. So this is just a disc diffusion assay where we're taking culture supernatant from the native PTT producer. We spot it on a paper disc with a lawn of Bacillus subtilis, and you get this zone of killing. Here's Streptomyces lividans, which doesn't make this antibiotic. Here's Streptomyces lividans that we put the large insert clone that came from this, and it's acquired the ability to make the antibiotic. All right, so we know that it makes it because it makes it. Here's a resistant mutant to PTT to show that this is not some other antibiotic that we've cloned on there, but it's actually phosphinothrycin tripeptide. We have another tool that I want to show you that we use widely for these studies, which is phosphorus NMR. So phosphorus chemistry is actually also fairly easy to analyze in the lab. The only stable isotope for phosphorus is phosphorus 31, and it's an NMR visible element. So you know there's a lot of phosphorus in cells. Phospholipid, you got ATP, GTP, DNA, RNA. All of those are phosphate esters. And in an NMR experiment, they have a chemical shift of 0 plus or minus 5. The carbon-phosphorus bond shifts these grossly upfield, anywhere from 15 to minus 45. So here you can see authentic phosphinothrycin tripeptide by NMR, and here's our heterologous recombinant strain, which has acquired the ability to do it. What's so nice about this technique is because these NMR peak is shifted so far from all the other phosphate esters, we could grow these in 50 millimolar phosphate buffer and have an organism that produced 5 micromolar phosphonate, and we can still see specifically the phosphonate, even though there's a huge background of other phosphorus compounds in the cell. This is a really powerful technique for us, and I'll show you how we use that. All right. So we have this kind of generic flow that we use for this. We get an organism that produces an antibiotic, we make the library, we screen it, we sequence the gene cluster, and one of the things that we would like to do is work out the biosynthetic pathway for each of these, and I'll show you in a minute why we would like to do that. So we do that by making block mutants. As I said, I'm a microbial geneticist. We also can take these individual genes and overexpress the enzymes in E. coli and do in vitro biochemistry to prove the proposed biochemical steps. In many cases, we've done crystal structures, which has turned up some really interesting biochemistry. We also use mass spectrometry and NMR to characterize these products. And in the end, we hope we get out of it a complete biosynthetic pathway. What I'm going to tell you is because phosphonic acids are kind of, did any of you ever study phosphonic acids in biochemistry? In chemistry? So, yeah, I'm not surprised. I haven't yet had anybody say yes to that question. You might imagine, though, because they're so different, that the biochemistry of these pathways is very different. And so in addition to learning the biosynthetic pathways for some strain engineering techniques that we want to do, we also have learned some really fascinating biochemistry. I want to give you an example of that. So one of the enzymes that we discovered in the course of working out this phosphinothrycin tripeptide pathway was this enzyme called PHPD. So in the process of making that antibiotic, it takes this intermediate, hydroxyethylphosphonate, and converts it to hydroxymethylphosphonate and formic acid. We know that because when we make a mutation in this, it accumulates this product. And here what I'm showing you is high-resolution mass spectrometry, proving that this is the actual mass, 4 decimal place mass accuracy. So that's the benefit of working with Neil Kelleher, who has some really high resolution mass spec instruments. If we take that enzyme and overproduce it and feed it this substrate in vitro, we get these 2 products, and here's some mass spec showing the results of that. So if you're good at biochemistry, you're looking at this reaction, and what you see here is cleavage of an unactivated carbon-carbon bond using molecular oxygen without input of exogenous electrons. That's an unusual biochemical reaction. It's actually an unprecedented biochemical reaction. We studied this a little bit, so we also determined the crystal structure of this enzyme. It's a member of a cupin family of known iron-dependent dioxygenases. And although you couldn't tell it from looking at the primary sequence of the gene, looking at the crystal structure, we can see that this PHPD enzyme, actually, the single polypeptide of this is actually a repeated subunit that looks like a dimer of this enzyme HPPE, which coincidentally catalyzes this unprecedented epoxidase reaction that's involved in production of the phosphonate antibiotic phosphomycin. So this was a previously studied by a guy named Ben Liu at the University of Texas. Also a very interesting reaction. in which it uses molecular oxygen to perform this epoxidation reaction. But here there requires exogenous electrons, and both of the oxygens end up in water. Here you don't need input of NADH, and both of the oxygens end up in the substrate, one on each of the carbon molecules. I'm going into this in some detail because I'm going to come back to this at the very end of the talk. So I want to show you this one because it's interesting and unusual biochemistry. Number two, because it's going to come back at the end of the talk, so I want you to remember this. Okay. So our goal is to work out a complete biosynthetic pathway. So here's our revision of the PTT pathway, which produces this glutamine synthetase inhibitor. So we've done this for lots of them. The question I haven't answered for you is why do we care about the pathways other than I happen to really like metabolism. So when I talk about metabolic pathways to my students, they kind of roll their eyes. I really love this stuff. This is really cool. So this gives me a chance to learn the metabolism that I really like, and also hopefully find some antibiotics that will be useful to people. So it's kind of a double plus. It's a win-win situation. Why would we like to know these pathways in such detail? If you know a pathway in incredible detail, you can begin to manipulate it in the lab. So lots of these antibiotics cannot easily be synthesized. Or if you can synthesize them, it's expensive. So if you think about the kind of problems there are with pathogens and drug-resistant pathogens, we in a rich country like the U.S. are fine. We can afford to pay, well, I don't know, maybe we can't afford to pay, but our HMOs typically do, high prices for antibiotics. But if you're in the third world, places like where malaria is a big problem, they can't afford really expensive antibiotics to treat malaria. 1 out of 5 people in the world has malaria. And if we give them expensive antibiotics, it's not going to do them any good. If we understand the way these are made, we can engineer the organisms to, instead of producing this much, to produce this much, and with a corresponding lowering of the cost of these antibiotics. And so this is a very powerful technique. Not only can we lower the cost of production of the knowns, once we understand these pathways, we can begin doing what's called combinatorial biosynthesis. So we can take the steps from one pathway and add it to another pathway and get new derivatives that nature hasn't discovered yet, or at least that we haven't discovered, nature's discovered. And maybe get novel activities that might be more toxic to the target organism and hopefully less toxic to human beings. So there's good reason to study these pathways in great detail. So it's not a coincidence that I mentioned malaria. This enzyme, FR900,098, I'll take you a minute to tell you what I think is a really cool genomic story. So this is an antibiotic that was discovered by Fujisawa Pharmaceuticals back in the late 1970s. It's a very good antibacterial agent. What it affects is the non-mevalonate pathway for isoprene biosynthesis. So all organisms need to make isoprene. Human beings use it to make cholesterol and other hormones, as well as quinones. Bacteria use it for quinone biosynthesis mostly, but also for other things that the cell makes. Bacteria make it by a completely different pathway than human beings. So because this targets this pathway, it kills bacteria, and because you don't use this pathway, you use this one instead, you can take literally grams of this stuff a day. It's utterly non-toxic to human beings. On the contrary, if you want to lower your cholesterol, you take statin drugs, which if you give to bacteria, they don't do anything. So this is an example of a biosynthetic pathway that's present in the target and absent in humans. So this antibiotic, it's a really good antibiotic. It went through human clinical trials. It's approved for human use, but it's expensive to make. And because we had lots of good antibiotics in the 70s and early 80s, it never found its way into widespread clinical use. Until they sequenced the genome of Plasmodium falciparum, which is the causative agent of malaria. And it turns out that that parasite has a bacterially derived organelle, the apicoplast, which uses this non-mevalonate pathway. And the malaria parasite is killed by FR900,098 with a 3-day course of treatment, including multi-drug resistant strains of malaria. So this is a very potent anti-malarial agent, which is now, because of genome sequencing and because the target was known, in phase 3 clinical trials in Africa as an anti-malarial. The problem is, it's too expensive. So fortunately, we worked out the genes that are responsible for its synthesis, and I'm not going to walk you through all this. We worked out the biosynthetic pathway. So here's an enzyme assay showing that we've got the clones that inhibit the target enzyme. Here's an NMR assay. Here's a high-resolution LC-MS assay. Here's a bioassay where we have a phosphonate-specific transporter. So they're sensitive to it anyway, but when you turn on phosphonate uptake, they become very sensitive to it, and that's how we know that it's a phosphonic acid. This is one of our discovery tools. Having worked out the biosynthetic pathway, we've cloned this into E. coli, and we now have engineered strains of E. coli that produce 6 times as much of this antibiotic as the original native host. Still not cheap enough to compete with chemical synthesis, which is still too expensive to find its way into widespread use. However, I would point out that other engineered strains have achieved million-fold increases in titer. And I think synthetic biology and strain engineering approaches, this is something that's definitely achievable. And we think we can make engineered strains that will make this antibiotic affordable for worldwide use. So that's why we would like to study the known pathways. So we've done this now for everything that's boxed in blue. So the technique works. Our genetic techniques are very powerful and allow us to work out these biosynthetic pathways. And it's an established technique for things that we know make phosphonic acids. The question is, can we use it now to discover new antibiotics? So I want to ask a series of questions. How common is phosphonate production in nature? Can we discover new natural products using this gene-based approach? And how many different kinds of antibiotics in the phosphonate class has nature discovered? All right, so we can answer that first question, how common is it in nature? Just by looking at sequenced genomes and looking to see if they have this PEP mutase gene. So it turns out that about 5% of microorganisms have the genetic capacity to produce phosphonic acids. Other than the Burkholderia, all Burkholderia make phosphonic acids. Other than the Burkholderia, this is not a phylogenetically coherent trait. So you can take any one of these different groups, and what you're likely to find is that about 5% of the members of that group have the genetic capacity to produce phosphonic acids. So it's dispersed amongst nature, but about 5% of genomes have it. But genome sequencing has not been a random sampling, so maybe that's not the best way to look. Maybe we should go to nature and isolate random microorganisms and ask how many of those have the genetic capacity. I told you at the beginning that streptomyces are very rich in antibiotic production. So we thought if we're looking for antibiotics, we ought to at least start by looking at streptomyces and the related actinomycete bacteria. So there's good selective enrichment for actinomycetes. We went around Champaign-Urbana, got soil, and we isolated them and we screened them for the ability to make phosphonates. The answer is 5% of randomly picked ones have the capacity to do that. We cloned and sequenced all of them. And what I'm showing you here is a phylogenetic tree of the PEP mutases that we found in those bugs, and just kind of an arrow diagram showing you the different gene clusters. And this isn't supposed to mean anything to you other than the fact that this line is different than that line, is different than that line, is different than that line. So all of these are different gene clusters. All of them direct the synthesis of phosphonates, based on the presence of the PEP mutase and other genes that look like they're doing small molecule biosynthesis. And I'll show you in the end that at least one of these produces bioactive phosphonic acids. The point here is after having sequenced 25 of these, none of them corresponded to the known phosphonates that we had looked at before. So we can discover new ones, and there's a fair amount of diversity here. So we were pretty encouraged by this. And it led us to ask how many different ones are produced in nature. So we didn't find any new ones, but we did find a bunch that were the same. In fact, by the time we'd looked at about 50 of them from Champaign-Urbana, half of them had the same gene cluster. So the question is, were we isolating them in the wrong way, or were we looking in too geographically constrained area? Maybe we needed to go around the world and look at lots of different actinomycetes that were harvested in lots of different ways, lots of different selective enrichments. I didn't want to do that. I mean, we could have done it, but that's a lot of work. So I contacted the ARS strain collection. So this turns out is the largest publicly held strain collection in the U.S. It's held by the Department of Agriculture, their Agricultural Research Service. And for us, it's convenient because it's in Peoria, Illinois, which is about a 70-minute drive from Champaign-Urbana. So it's a huge strain collection. They have almost 10,000 actinomycetes in their collection. We worked out a material transfer agreement, and we have a replica of the entire actinomycete culture collection. This culture collection is over 8,000 isolates from 115 different countries and includes 116 different genera within the actinomycetes. So I have a very talented postdoc who worked with Becky Perels, who's sitting in the audience here today. His name is Kusan Zhu. My longtime technician, Junkai Zhang, and three very talented undergraduates, John, Jolene, and Emily. In 9 months' time, these 5 people got in 9,000 lyophilized strains. They revived all 9,000 strains and grew them up in culture. They saved all of them in the freezer. They prepared genomic DNA from all 9,000 strains, tiled the genomic DNA into 96 well plates, screened them with our 4 pep mutase primers. That's 35,000 PCR reactions. They did all this in about 9 months. It blows me away. This is entirely due to Kusan Zhu. He's a very talented organizational guy. He's extremely hardworking. And then a group of really good, talented undergraduates who were just into the project. So out of that, we got 346 strains that contain the pep mutase gene. They have the genetic capacity to produce phosphonates. In the next 3 months, they then grew up all those 346 strains, prepared cell extracts, and ran them through a phosphorus NMR and a mass spectrometry screening platform to identify which ones had the genetic capacity, I'm sorry, not the genetic capacity, which ones expressed their genetic capacity to actually make phosphonic acids. About 15% of them. Now I should tell you that the way we do this NMR screening, it takes about an hour per strain. And we're being charged $16 an hour to do this. So we looked at one culture condition and one time point, and we got 15% of them producing phosphonic acid antibiotics. I think later on we can go back and screen these in different culture conditions and using a variety of genetic and other medium manipulation tricks and get some of those remaining 85% to also produce phosphonic acids. So this is just a very first pass, kind of a low-hanging fruit screen. So we wanted to then identify the biosynthetic gene clusters that were present in these. I showed you our phosphid library technique. That used to be the most efficient way to do this. But as many of you know, genome sequencing technology is coming down really dramatically. So when we would do the phosphid cloning, that would take us about 3 weeks' time, and it would cost us somewhere between $1,500 and $2,000. With Illumina sequencing and a sample pooling platform, we can sequence the genome for $200, and all we have to do is hand the genomic DNA to our sequencing center. So it's faster, it's about 8 times cheaper, and not only do we get the gene clusters that we're interested in, we get everything else that's in the genome as well. So in the last 2 months, we've sequenced 100 streptomycete and other actinomycete genomes and pulled out the phosphonate biosynthetic gene clusters from each of those. I'm going to talk about that in just a minute. But the other thing I want to tell you is that in addition to discovering those 100 phosphonate gene clusters, keep in mind that every one of those genomes has the genetic capacity to produce between 20 and 40 other natural products. So in that sequencing reaction where we got our phosphonate gene clusters, we've also discovered the gene clusters for somewhere around 3,000 other natural products produced by actinomycetes. So we're not going to follow up on that. But that's a wealth of data for the natural product field to take advantage of, and is going to help people uncover the biosynthetic pathways of other natural products. So that's kind of a bonus that we get from this genome sequencing approach. So the goal of this was to find out how many different kinds of phosphonates are produced. So we did a gene clustering algorithm. What we're simply looking at here are the different gene clusters, and everywhere there's a line drawn between them, 30% of the genes in the gene cluster are shared in common. So these don't necessarily produce the same products, but you can see here's a big cluster that probably produces a similar molecule, There's another similar molecule. So out of these 100 gene clusters, we expect to get something like 50 or 60 different phosphonate natural products, which, based on what I told you at the very beginning, are very likely to be bioactive. The phosphonate gene clusters, the pet mutase gene itself, shows a great deal of diversity. As these color codings show, we can predict partial pathways for a lot of these. Many of these are really useful. So here's that threonine synthase inhibitor that I showed you at the very beginning. This is an interesting molecule. It's actually made in 2 different flavors. One is called rhizocticin, and it's a very potent antifungal agent. The other one is called, and has no activity on bacteria. The other one is called plumbamycin. It's a very effective antibacterial agent. Has no activity on fungi. The way they work is they produce this molecule, APPA, which is the threonine synthase inhibitor. The way they're targeted to the microorganism is they're made as a tripeptide. And the amino acids that are hooked onto the warhead of the molecule are different between these two. And what that means is it's the amino acids of the peptide that tell you the target organism. And so by changing these amino acids, you can target this antibiotic to different groups of microorganisms. Well, it turns out that in addition to these two, we have at least 4 other gene clusters which make the warhead, but have different peptide synthases to attach different amino acids. So here's an example where, by just doing the genome sequencing, we're not really discovering a new antibiotic, but we're discovering the potential to target other groups of microorganisms. It turns out this is that antimalarial gene cluster. We wouldn't have realized it by sequencing it because the genetics told us that these genes alone were enough to make the antimalarial. But by seeing the conservation of the genomic region, it also happens to be next to some peptide synthases. And we suspect that these organisms now, in addition to making that antimalarial molecule, also make that antimalarial molecule conjugated to some amino acids, which potentially has implications in targeting and host specificity of that antibiotic as well. So we're actively following up on that. These 6 produce unique phosphorus NMR signatures. All of them are bioactive against a panel of test strains. And this is that phosphonate uptake strain, and the bioactivity is enhanced by doing phosphonate uptake in 4 of the 5 strains that we've tested here. So we clearly have discovered, based on the gene clusters, novel phosphonic acids with antimicrobial activity. Like I said, we only did this in the last couple of months, so I can't tell you what these molecules are. We're actively purifying them, and we'll determine the structure. We'll work out the biosynthetic pathways using the techniques that we've discovered up till now. So I think this gene-based approach is a really powerful approach that's going to allow us to uncover new, novel bioactive molecules. We've also learned a lot of novel biochemistry along the way. And I'm going to show you with the remaining time I have left that although we were looking for antibiotics, we found things that go maybe a little bit outside the antibiotic discovery field. So as Dave told you in the introduction, I'm also very interested in methane, and I do a lot of work in my lab on methanogenic archaea, totally unrelated to this project. People for years would ask me as I go around, is there any connection between the two? And the answer is no. But I'm going to show you that actually by studying antibiotics, there is a connection between these two, although it's not actually what I expected. So here's some studies from the late 1970s showing an interesting phenomenon. So I'm really changing views here. This is a profile of the methane concentration in the open aerobic ocean. And as you go down in depth to around 100 meters, there's a sharp rise in the concentration of methane, which as you go deeper falls off again. So if you look at these profiles, if you're an environmental microbiologist, when you see a spike like that in concentration, you know what that means? Anybody? It means production. So there's a source of methane in the aerobic ocean. So anybody who's studied methane, biological methane production knows that this is done exclusively by a group of strictly anaerobic members of the domain archaea. So no bacterium, no eukaryote has the capacity to do this. And the organisms that do this are highly sensitive to oxygen and can't make methane in the aerobic waters. So if you look at this, there's a lot of methane here. In fact, it's super saturated with methane relative to the concentration of the methane in the atmosphere. And it's also completely saturated with oxygen. So this can't possibly be caused by anaerobic methane-producing archaea. And yet there's a clear source here, and the amount of methane is substantial. This is about 4% of the global methane budget. Methane is a very potent greenhouse gas. It's a major contributor to climate change. If there's something that's about 5% of the methane budget, and we don't understand where it's coming from, that's probably something that's worth looking into. So this is known as the ocean methane paradox. How can you get methane production in the aerobic ocean? So Dave Carl and his collaborators came up with a model for this. They suggested that this phosphonic acid, methylphosphonate, when it's acted upon by an enzyme called carbon phosphorus lyase, would reduce phosphate and methane. And because it's known that phosphonic acids, and I mean phosphonic acids in general, not methylphosphonic acid, are abundant in the ocean, and because the ocean is phosphorus limited, then it would be to the advantage of organisms that live in the ocean to take use of this methylphosphonate, which they hypothesized would be present, and in the process of doing so, they would release methane. Right. So that's an interesting model. And when I talked to one of the co-authors, I told him that that was complete and utter, and you can fill in the blank. I won't use it in a seminar. I thought this was ridiculous. And I thought this was ridiculous because this is actually something that I've worked on since I was a graduate student. So I did my PhD thesis on this enzyme carbon phosphorus lyase. This is not novel. This has been known for 30 years, so actually 40 years. In 1972, a scientist named Doughton showed that organisms that have carbon phosphorus lyase, if you feed them methylphosphonate, will produce methane as the other product. So that's not surprising. It's known that the organisms in the open ocean, because it's phosphorus limited, commonly have carbon phosphorus lyase. Yeah. So this is well-known biochemistry. So this is my PhD thesis. These are two organisms that I worked on subsequent to that. This is something that I've worked on for a long time. So the problem with the model is that there is no such thing as methylphosphonate in nature. We used to buy it from Signal Chemical Company. And that's the only source that I knew of for methylphosphonate. More than that, I've showed you in the previous part of the talk. We spent a lot of time working out biochemical pathways for the synthesis of phosphonic acids. No known organism makes methylphosphonate. And we know a lot of intermediates and a lot of pathways. None of those intermediates is methylphosphonate. And none of those intermediates could be converted to methylphosphonate by known biochemistry. And in fact, in order to cleave some longer chain carbon compound and produce a methyl group there would require unprecedented biochemistry. And that was the reason I said that it was complete and utter nonsense. It's a nice model, but there's no known natural source for methylphosphonate. So that can't be the source of methane if that doesn't exist in nature. So we've been studying antibiotic biosynthesis for quite a while. I'm interested in archaea. So it turns out there's an organism. This is a phylogenetic tree of known PEP mutases. Turns out there's an organism called Nitrosopomonas murilis. Maybe some of you know this organism. This is something that Dave Stahl isolated. It's an ammonia oxidizing archaean that's highly abundant in the oceans of the world, where it's involved in nitrogen cycling. It has a PEP mutase gene cluster. We can actually recognize a lot of these genes. So having worked out a lot of these pathways, we knew that it had a PEP mutase, it had a decarboxylase, it had a dehydrogenase, so it certainly would make this substrate. And it had an enzyme that had weak homology. Now, go back to that enzyme I told you to remember, because we were going to demonstrate it again. That would cleave this unactivated carbon-carbon bond and produce hydroxymethylphosphonate and formic acid. Very weak homology. If you did a blast search, it would return a value of about e to the minus 5. So most of you would consider that to be a marginal hit. But we thought it was close enough that and the biochemistry of that enzyme was interesting enough that we wanted to overproduce the enzyme and test its activity in vitro. So we did that. So we fed it C13 labeled hydroxyethylphosphonate, and we looked for the production of hydroxymethylphosphonate, and that's not what we saw. So here's phosphorus NMR. This is the substrate. So we spiked in the substrate, and you can see that this is a different NMR signal from that. That signal does not correspond to hydroxymethylphosphonate, and in fact it corresponds to methylphosphonate. So I admit I was wrong. Methylphosphonate can be a natural product. It does involve a really unusual enzymatic reaction, which involves cleavage of this unactivated carbon-carbon bond to give methylphosphonate. So we've shown this by C13 NMR, by phosphorus NMR, by high-resolution mass spectrometry. In fact, we've crystallized the enzyme and we have the structure of it. And interestingly enough, it's of this same enzyme family. But here's a really unusual enzyme family. In one case, it catalyzes epoxidation, and the oxygens end up in water. In another case, it catalyzes cleavage of this bond, with one oxygen ending up there and another oxygen ending up there. In the second case, it cleaves that bond again, but in this case, both of the oxygens end up on the terminal carbon, and it produces carbon dioxide, or bicarbonate, at the pH that we do this reaction. So it is a natural product. It is produced by an unprecedented biochemical reaction, and it's present in an organism that's known to be highly abundant in the aerobic ocean, Nitrosa pumilus, which Nitrosa pumilus and its relatives have been estimated to have a global population of something like 10 to the 27th microorganisms. Is it 10 to the 5th per mil throughout the entire world's oceans, something like that?
Between 10 to the 4 and 10 to the 5.
So there's lots of them. So if they make this compound, they presumably are the source of methane in the aerobic ocean. They're not making the methane themselves, but they're putting carbon into this compound, which when the Nitrosa pumilus cells die, the other phosphorus-starved organisms in the ocean take it up and use it, and are probably responsible for that methane spike in the ocean. So it gets just a little bit more interesting. Dave and his postdoc Willem, I guess Willem's in there, he's there in the back, grew up Nitrosa pumilus cells. We can show in vivo that cell extracts actually make phosphonic acids. So these are the phosphates that I told you about. And up in the phosphonate range, you see that they clearly produce phosphonic acids. Based on the gene context of that gene cluster I showed you, they probably make an exopolysaccharide or a glycoprotein that's decorated with methylphosphonate. So as I said, it gets a little bit more interesting, because now that we know the gene that's responsible for the synthesis of methylphosphonate, we can go out and look in metagenomic data sets and ask how common is it in the ocean. Well, we actually found almost 70 examples of it in the Global Ocean Survey data for this big metagenomic data set that Craig Venter put out a few years ago. You can look at the scaffolds that these are found in. And so here's the Nitrosa pumilus gene cluster. And you can see that it has some of the genes common next to it. You can see this sulfatase gene, the dehydrogenase, the decarboxylase, the PEP mutase gene. And in one case, it's immediately adjacent to a 23S RNA gene. And so we can identify the organism that this came from. And this came from an organism called Pelagibacter, which is the most abundant microorganism in the ocean. So now we find it in Nitrosa pumilus and also in Pelagibacter. So 2 out of the 3 most abundant organisms in the ocean. We'd like to know where these come from. So there's a technique you can do called phylogenetic profiling, where you can take short DNA reads and run it through a computer program, and based on a training set, it'll predict what organism it came from. So the people who developed this software will tell you that it's 90% accurate at predicting the phylogeny of these DNA sequences. You can believe it or not. I don't want to vouch for it, but it's an interesting idea, and if you do that, you can look and see what organisms you see. And you see Nitrosa pumilus, you see Pelagibacter, and you see Prochlorococcus. So Prochlorococcus is the 3rd most abundant organism in the ocean. And so what we've gotten out of this antibiotic study, walking us unwillingly into microbial ecology, is that the 3 most abundant organisms in the ocean produce methylphosphonate in exactly the same zone where you see this paradoxical paroxysmal methane peak in the aerobic ocean. So that's kind of a nice story. We certainly have good data for this one. We have the 23S data for this one, and if you believe phylogenetic profiling, we have Prochlorococcus represented as well. So that kind of took us off in a different direction, and I just think it's a nice example of how we set off to discover novel antibiotics. I really believe in basic research as a way to discover things that aren't necessarily what you're looking for in the first place, but which may be of global environmental significance. And I think this is a really nice kind of serendipitous example of that. So let me just finish with this. Phosphonate synthesis in the ocean is really abundant. We find it's something like, you can look at the occurrences of the methylphosphonate synthase, you can look at the occurrences of the PEP mutase gene. They're about at 30% of the phosphonate synthesizers might have the capacity to produce that. And if you compare these numbers to other single copy genes, we find that somewhere between 4 and 12% of all microorganisms in the ocean synthesize phosphates. We've done similar studies in soil metagenomes. It's very abundant in soils as well. But it's not the same PEP mutases. It's a completely different set of PEP mutases. And what this is telling you is that there's a huge diversity of phosphonate synthesis in nature. These are really abundant molecules. It turns out that phosphorus is the limiting nutrient in almost every ecosystem on the planet. Here's a class of phosphorus-containing molecules that's largely been ignored by people who study biogeochemical cycles, which clearly is highly abundant based on gene signatures, based on measurements that have been taken in a few cases, and I think is a really underappreciated class of molecules in nature. So that's how we got from microbiology into phosphorus cycling in nature. And that's where I'm going to stop today. So I just want to acknowledge the people who did this, particularly Kusan Ju and James Doragasi, who's done all the bioinformatics of our genomes. As I said, I collaborate with Neil Kelleher's mass spectrometry group, Wilfred Vanderdank's chemistry group, Huimin Zhao's strain engineering group, Satish's protein crystallography group, and of course Dave Stahl and Willem, who've helped immensely with our ocean methylphosphonate study. And that's where I'm going to stop.